Hello, this is Joseph Kazari at Mississippi College. I am the lab coordinator and instructor for several classes, and this lecture is for Bio 111 Lab, Experiment 5, I believe it is. And we're going to be discussing, you will be discussing this week, working with cellular respiration. A couple things about cellular respiration. First of all, anytime we hear the word respiration, we're dealing with the exchanging of gases, or maybe even the production of gases. Now, you breathing is respiring, but in your cell, when they exchange gases, or converting, changing, that's referred to as cellular respiration. Cellular respiration is when you have a sugar, like glucose, C6H12, O6, and oxygen, so there's our gas right there, and through a series of reactions, our arrow means yield, will form carbon dioxide and water. Now, to balance this, we're not too worried with balancing, but we are producing numerous amounts of carbon dioxide and water from just one glucose molecule. Now, carbon dioxide and water are just the byproducts of cellular respiration. Because while this is forming, in the process of making this, we are taking a substance called ADP, and we are charging it up. In other words, we are adding a phosphate to it. We're going from adenosine diphosphate to adenosine triphosphate. And this is the energy we want. And from one molecule of glucose, we get approximately 36 ATP. And this may vary from text to text or publisher to publisher. Well, since we're using oxygen here, right, in this reaction, cellular respiration is an oxidative process. Right. Same thing as if you have iron and the oxygen reacts with it. That is an oxidative process. It is oxidizing another substance. Cellular respiration occurs in three phases. The first phase, well, let's say three phases. The first part is glycolysis. The second is referred to as the citric acid cycle. It may be also referred to as the Krebs cycle. But that's kind of been phased out a little bit, but you still see Krebs cycle in random books. And the last phase is the electron transport chain. One, two, three. Now, with these three, it is a very good idea to know where in the cell each one of these are occurring. And this is going to help you with your biology as well, because this is not going to go away. Very important process, cellular respiration is. Right. Where is glycolysis happening? Citric acid and the electron transport chain. What does it react on? All right, in other words, what starts off in glycolysis and what do you end up with? All right, so what's the reactants, what's the products? Citric acid cycle. Right, you should know what's the reactants, what do you start off with, what's the products? Right, and same thing with the electron transport chain. What are the reactants, what are the products, and where they are. And we're going to go through that briefly in a moment. Now, in terms of respiration, there are two types of respiration. Right. Cellular respiration, for the most part, we have oxygen. We refer to that as aerobic respiration. Whereas some conditions, some organisms live in an environment that's void or lacking oxygen, but somehow they still produce energy. Even in our body, when we are short oxygen, we can utilize some anaerobic respiration. Okay. For starters, let's talk about the three phases. And we're going to be using oxygen, so we're talking about the anaerobic process, right? Aerobic respiration. We'll talk about anaerobic in just a moment. So let's see. Here's our cell. Now this will not be true to scale. Here's our cell, and here, right, cell, and here is a mitochondria. Mitochondrium. Of course, this is not true to scale, because in a cell you can have anywhere from 1,500, 1,700, 1,800 mitochondria inside of them. But this bean-like organ has two main compartments. It has this fluid inside the mitochondria, and it has these folds. I point those out because those are going to be important in a moment. And inside the cell, yes, we have other organelles, but that's not important right now. Right? What's holding and housing all those other organelles is cytosol. All right? So let's start off with glucose. Glucose is a 6-carbon molecule. Now, just for the sake of time, right, I'm going to leave off hydrogen and oxygen. We still need to understand that oxygen and hydrogen are there. So we have this 6-carbon molecule. This 6-carbon molecule, by a series of reactions, will break down to 2 3-carbon molecules. Now, if this was glucose, all right, we have these 2 3-carbon molecules is pyruvates. All right, maybe referred to as pyruvic acid. It's okay. What we just did is we took a 6-carbon molecule and we split it. The name up here, glycolysis. Anytime you see glyco, that's referring to sugar. That should be a good indicator right there. That's a context clue. Lysis means to split. And that's exactly what we did. We just split it in half. Now, there are some things that are produced here that we're going to skip over for right now. There's a little bit of ATP produced. But at this step, after glycolysis, notice that we're inside the cytosol, the cytoplasm. Right? The pyruvate will then migrate into the mitochondria. So glycolysis, right, we're in the cytoplasm, cytosol, cytoplasm. It both works. All right, and we produce pyruvate. The pyruvate is going to go into the mitochondria, not just anywhere. We have these folds, and we have another substance in here. The substance is referred to as the matrix. So the next step, the citric acid cycle, is going to occur in the mitochondrial matrix. And it is a cycle. What happens here? we are skipping over some items. You'll go into much more detail in your Biology 111 class. I'm just going to say CAC, citric acid cycle. These 3 carbon molecules come in, and what they're doing is they're taking off carbon and oxygen, so carbon dioxide. They're producing some ATP. Since I said that here, let me say it here. Some ATP. But what's left? Our carbon that we started off with, the glucose, is carbon, hydrogen, and oxygen. So all we did was separate it. And then in the cycle, we are getting rid of carbon, oxygen. What's going to be left? All right, that's going to be hydrogen. That hydrogen, it's not just hydrogen, it's also electrons. Let's see, so this hydrogen and electrons, and we're just going to make it move right over here. Hydrogen and electrons are going to go to these folds. You can go anywhere. I'm just trying to make some distance here. And that's going to go to the, we call that the Christie. There's a lot of proteins in that inner membrane, just like there's a lot of proteins in the cell membrane, where the electrons are going to be used as energy, and the hydrogens are going to be moved around, and this is where we're going to, this is the location where we are going to make a lot more, the majority of our ATP in the electron transport chain at the Christie. But when that hydrogen is all said and done, notice that we just used these substances. We used the glucose. We produced carbon dioxide. We didn't even use our oxygen yet. When this hydrogen, these electrons are done, well, we need an electron acceptor. We can't just leave these things hanging around. That may cause some problems. This is where the oxygen comes in. And it's going to react with it. The hydrogen, electron, and oxygen, right there we use our oxygen. And it's going to hold on to it. It's going to bond with it. It's going to form a compound water. So what we have here in the electron transport chain found in the Christie, right, we're using oxygen and hydrogens and electrons to produce water. And that is cellular respiration, what happens inside you, inside of a nutshell. That was done in less than 10 minutes. And you're going to hear about it again and again and again and go into much more detail with your following classes. So if we're doing a lab on this, what we're really doing is we are confirming that this is going to happen. So how can we measure it? Well, let's see. We can measure it by, if we could figure out how much oxygen is used. Right, if we know how much oxygen gets sucked up into some cells. We can measure it by, right, since we're making carbon dioxide, right, that's a product, how much carbon dioxide is produced. All right, that looks pretty easy. And there are some tests out there. Maybe we can determine how much energy, ATP, was released. But that would cause us to spend a lot more time discussing how that is and the actual tests. So it's not very easy or practical way for this lab. So we are going to do both of these. We're going to be measuring how much oxygen is used for cellular respiration and how much carbon dioxide is produced. How we're going to do that is in procedure 3, right, you're going to have what we call a respiratory tube. Your little glass tube, little metal disc, a washer on the bottom. All right, we've got a rubber stopper, right, with a little pipette here. So what we will do is we'll use our respiratory tube. We're going to place some cotton in the bottom of it. This is going to be absorbent cotton. Then we're going to put another type of cotton on here, which is non-absorbent cotton. Now we're going to do this because we need to add something else. Well, let me go ahead and finish this up. We're also going to be putting some peas, a certain number of peas. You got to figure out how much volume those peas take up. Because even though these are plants, they are producing carbon dioxide just like we do. And they're also using oxygen. But what we're going to do is we're going to measure how much oxygen is used. In fact, let me write this a different. Let's say this. These peas will produce carbon dioxide, but they're also going to use oxygen. What we're going to do is put a rubber stopper on the top. We're going to put that micro pipette there. And we're going to lay this down in some water. Make sure the top is sealed. And when it gets in the water, if the oxygen is being used, water will be moving up this micro pipette, or this pipette. It's a 1 millimeter pipette, excuse me. But the problem is, if carbon dioxide is produced by these peas, which they should be, that would push it out. So there's a problem. We don't want to know how much carbon dioxide is produced. We only want to measure this for this procedure. So what we do to stop that is in this absorbent cotton down here, we're going to put potassium hydroxide, which is a base. Potassium hydroxide, aqueous, it is going to react with the carbon dioxide gas that is produced in this chamber. And it will produce potassium carbonates. It's an aqueous. I think your book might also say a solid, but I mean, if it's wet, it's going to be an aqueous. And it's going to produce moisture, water. But notice here, by this reaction, we're not producing any gas. So by using potassium hydroxide, we are eliminating that carbon dioxide. When you put this tube into the water, you need to be careful what you're looking at, because if you look at one side, it has the volume increasing as you move down. We're not looking at the large bold numbers. We have the small numbers on the other side. We're going to be looking at as water is moved up. And this is what we see here. It may be difficult to see, but you're going to have to try to use your imagination. You'll see it when you get in lab. All right, if all of this is 1 millimeter in this pipette, we have 0.1 of a millimeter, a tenth of a millimeter, 0.2 of a millimeter. If water is being sucked up into this pipette, that's telling you how much oxygen is being used. You need to be sure you're reading the correct part. Right? So if this is a tenth of a millimeter, Of course, we go in between there, so the middle line would be 0.15 of a milliliter, 0.16, 0.17, 0.18, 0.19, and so on. So of course our little marks here are hundredths. All right, just be sure you're reading that properly, and do not take it out of the water when you measure it. Every time you pick it up out of the water, just moving it up, all right, it's going to change the pressure, and your bubble may change. It will change. But in a good experiment, you don't do that just once. You're going to be doing this 2 more times. You need a control. You're going to be doing the same thing for some peas that, well, these peas right here, right, they are germinating peas. In other words, they're growing, they're respiring, they've been soaking overnight, and they are awake and ready to do their job. You're going to do it again with non-germinating peas, which are dry and have not been soaking. But you want to make sure you have the same volume. If this was 30 milliliters of peas, then you want to make sure you have 30 milliliters of non-germinating peas. And since they will not be as large, you're going to have to add some glass beads. So this is actually a control for volume, just so we know that it's the germinating peas that cause the oxygen, not the non-germinating peas. And then you're going to do it all, when I say again, you're going to do it all at the same time. You will have 3 of these respiratory tubes. Again with glass beads, which, beads, yeah, beads, I was going to say peas, which is your control for anything else that you may not have realized. All right. And the last thing which you will be dealing with, oh, by the way, you don't need a lot of cotton. Okay, just a little bit. You have 2 layers. Since this is non-absorbent cotton, this potassium hydroxide should not be able to creep up and affect our peas there. We want to keep them separate. And you don't want to have this, get this this potassium hydroxide on your skin. It is very caustic. So there's your aerobic respiration. Now let's briefly discuss anaerobic respiration. Anaerobic means without oxygen. So therefore, it is a incomplete respiration. For example, what you're going to be looking at today is you're going to have some glucose you will use. And we are going to use yeast. We need some type of living thing for aerobic respiration. So that's where we use the peas. Now for this, we're going to use yeast as our source of anaerobic respiration. But it needs the sugar. Now for this, we will not be adding oxygen. There's no oxygen there. Usually if it's underwater, there's not a lot. So what will happen is it will produce carbon dioxide and 2 C2H5OH, and it's going to produce some ATP. This produces energy because it is a type of respiration. C2H5OH, this is a waste. All right, this is what we want, the ATP. But this waste is ethanol. Some companies use anaerobic respiration as their livelihood. For example, let's say the beer and the wine industry, what they do is they will take, well, some sugars. Let's say we got some carbs, carbohydrates, maybe some fruits, because that's how you make wine. All right, it might be some grains. There's carbohydrate and grains. That's how you make beer. And they will add it with yeast. And the yeast is going to make our ethanol, which is an alcohol, which is drinking alcohol. This is what some other companies use. They use corn to produce alcohol so that we can use it in our gasoline. It's supposed to be able to burn cleaner. The baking industry. What they do is they use yeast. They're using the carbon dioxide. Now, yes, carbon dioxide is also produced over here. Right? They're using the same process with their breads. Now, they're producing carbon dioxide and alcohol, ethanol, to be exact. And this is going to help the bread rise or the cake rise. The alcohol just gets burnt off, just evaporates. So when you cut open your bread, you may notice that it has a lot of bubbles in it. Those bubbles are the carbon dioxide, which helped lift the bread. Now inside of us, we don't use yeast. Right? Our cells can use anaerobic respiration if we did not have enough oxygen. But what we would do is we would produce lactic acid and some ATP. Now, it won't feel good the next day, but usually when you run short in oxygen because you've been wearing yourself out, exercising, running, etc., you might have a little burst of energy, right, which is usually this anaerobic respiration, where using our sugars, we're producing carbon dioxide, and instead of ethanol, it's lactic acid. Today, you're also going to be using, for the anaerobic respiration, is these little tubes called a fermentation tube. Anaerobic respiration, also known as fermentation. Right. Usually when you hear about people fermenting things, you know, sometimes people apply it to alcohol. All right, but what you're going to be doing is you're going to be using yeast. You're going to put some yeast in here. You're going to put a certain amount of water. They'll show you in lab how to do this. And you may be, it might not just be water. It might be sugar water. So let's see. We're going to have some carbs. We're going to have some yeast. So we're going to be producing two things. And you're going to smell what it smells like. All right? You're going to mix it up when it's all in there. You want to fill this tube all up with water. We are going to measure, I should say, the carbon dioxide produced. You're going to set this fermentation tube in a beaker. If you set it up like this, then you can imagine where the carbon dioxide is going to go. It's going to leave. So when you set it up, try to let the neck of it here, this tube, which is going to collect the carbon dioxide, make sure that it is up and not at an angle. goes like this, not like that. Right. And last thing is this is week 3. Right. This is week 3 of our sea fern experiments. What you need to do is you need to observe your gametophytes. Observe and count your gametophytes. And don't forget to add some water. You can see the instructions on page 32, and I think you're going to write your information on page 35. I might be backwards. So refer to those pages of your lab manual. All right? And this is Joseph Gazzari for Bio 111 signing out.